Bismillahirrahmanirrahim. Starting with the name of Allah, who is the most beneficent and most merciful. This is Muhammad Ishaq, student of BS MLT, 8th semester, Cohort University of Science and Technology. And the topic I am going to present as anticoagulant. तो दोस्तों आज हमारा एक जो टॉपिक है वो एंटीकोगुलेंट्स के नाम से ही जाना जाता है। एंटीकोगुलेंट एक ऐसा कीमियाई मादा है जो कि लेबॉर्ट्री में खून के साथ शामिल किया जाता है ताकि ये खून ब्लड जो होता है वो क्लॉट ना हो जाए, वो जम ना जाए। आउटलाइंस: एडीटीआई, इथिलीन डाइअमाइन टेट्राएसिटिक एसिड, हेपारिन, सोडियम साइट्रेट, सोडियम फ्लोराइड। Anticoagulants, commonly known as blood thinners, are chemical substances that prevent or reduce coagulation of blood, prolonging the clotting time. एंटीकोगुलेंट जो आमतौर पर खून को पतला करने वाले के तौर पर जाना जाता है वो कीमियाई मादा है जो खून को जमने से रोकता है। Most tests performed in the hematology laboratory involve anticoagulant. So it means that हेमाटोलॉजी लेबॉर्ट्री में किए जाने वाले ज़्यादातर टेस्टों में एंटीकोगुलेंट खून शामिल होता है। Once the blood has left the body, a series of chemical reactions occur, causing blood to clot within minutes. एक बार जब खून जिस्म से निकल जाता है, रद्द-ए-अमल का एक ऐसा सिलसिला होता है जिसकी वजह से चंद मिनटों में खून जम जाता है। To prevent coagulation from occurring, a substance called an anticoagulant is mixed with the blood. तो इस अमल को रोकने के लिए, मतलब कि खून को जमने से रोकने के लिए एक ऐसा मादा जिसको एंटीकोगुलेंट कहा जाता है वो खून के साथ शामिल किया जाता है। टाइप्स ऑफ एडीटीए एंटीकोगुलेंट्स। टाइप्स ऑफ एंटीकोगुलेंट्स। फर्स्ट, एडीटीए. एडीटीए is the most commonly used anticoagulant in the routine hematology laboratory. एडीटीए रूटीन हेमाटोलॉजी लेबॉर्ट्री में सबसे ज़्यादा इस्तेमाल होने वाला एंटीकोगुलेंट है। एडीटीए प्रिवेंट कोगुलेशन बाय चेलेटिंग कैल्शियम, a necessary component of coagulation. So the meaning of this sentence is that ADTA combined with the calcium that is the necessary component of the coagulation process and so prevent the clotting of the blood. Its removal inhibits the coagulation process. It means that when the calcium is removed, then the coagulation process cannot proceed. मतलब ये हुआ कि अगर एडीटीए जब कैल्शियम के साथ कंबाइन हो जाता है, उसके साथ बॉन्ड बना लेता है, तो जो कोगुलेशन का प्रोसेस होता है वो स्टॉप हो जाता है और खून जमने से रुक जाता है। इथिलीन डाइअमाइन टेट्राएसिटिक एसिड is an amino polycarboxylic acid. This white water soluble solid is widely used to bind to iron and calcium ions. मतलब कि ये जो केमिकल है ये अक्सर आयरन और कैल्शियम के साथ बॉन्ड बना लेता है, वो उसके साथ कंबाइन हो जाता है। It binds these ions as a hexadentate chelating agent. EDTA is produced as several salts, notably disodium EDTA, sodium calcium EDTA, and tetrasodium EDTA. EDTA is used extensively in the analysis of blood. It is an anticoagulant for blood samples for CBC, complete blood count, or FBCs, full blood counts, where the EDTA chelates the calcium present in the blood specimen, arresting the coagulation process and preserving blood cell morphology. So it means that EDTA is mixed with the blood sample that we are using for the test of complete blood count or full blood count or preference smear or RBC morphology, etc. And by the chelating calcium, the EDTA arrests the coagulation process. Tubes containing EDTA are marked with the lavender or pink top. तो जिस ट्यूब में EDTA होता है उसका जो टॉप होता है वो या तो गुलाबी या लेवेंडर कलर का टॉप होता है, मतलब उसका जो कैप गुलाबी या लेवेंडर कलर का होता है। दी ऑप्टिमल concentration is 1.5 milligram EDTA per ml blood. Sample collection tubes can contain one of three different sorts forms of EDTA: disodium, dipotassium, tripotassium, dilithium EDTA. Testing Tests using EDTA samples include CBC, peripheral blood smear examination, reticulocyte count, and immunophenotyping of cells by flow cytometry. So it's mean that the tools with the EDTA that we use for the tests are CBC, peripheral smear, reticulocyte count, and immunophenotyping of cells. These are the tests for which we use EDTA blood samples. Ideally, the sample should be used within 6 hours of collection for the majority of these tests. So it means that after collection of the blood, the test should be performed after 6 hours, within 6 hours. The sample stability can be extended to 24 hours with storage at 4 degrees centigrade for certain tests such as the CBC. It means that we can extend the stability of these tests by keeping the sample at 4 degrees centigrade. If peripheral blood smear are to be prepared, they should be made within 3 hours of collection. So, if we collect blood in the EDTA tube for the peripheral blood smear, then the smear should be prepared within the 3 hours of the collection. After 3 hours at room temperature, degenerative changes can be observed by examining a red stained blood smear. False interpretation. Excess EDTA. For example, more EDTA, you feel less blood volume, so EDTA is in excess. It means that if the tube is filled for 3 or 4 ml, 3 or 4 cc blood, and you put less amount of blood and a tube, so it means that you fill less blood with more EDTA. Did cause shrinkage of RBCs, causing false erroneously reduced hematocrit HCT. Subsequent increase in MCHC, decrease in MCV, platelets are also affected. They will swell and subsequently disintegrate. Causing erroneously high platelet count, platelet will disintegrate into more than one fragment. Each fragment will be counted as one platelet. And now we are discussing the second type of the anticoagulant, sodium citrate. Sodium citrate is another anticoagulant that is used in the laboratory to be added with the blood to prevent blood clotting. Sodium citrate is the recommended anticoagulant for coagulation studies. The Clinical and Laboratory Standards Institute (CLSI) recommends the use of 3.2% sodium citrate. The sample collection tube stopper is color coded in light blue to indicate the presence of sodium citrate. Sodium citrate prevents coagulation by handling calcium in soluble complex. As the anticoagulant of choice for coagulation and platelet function test, also is used for ESR, erythrocyte sedimentation rate test. It acts by precipitating calcium, thus it will not be available for clotting process. Like EDTA, citrate acts by removing calcium from the blood. Unlike EDTA, it's reversible, so calcium can be added back to study coagulation under controlled condition. Citrate plasma is also used to measure coagulation relevant factors. And this is the third type of anticoagulants, heparin. Heparin is another anticoagulant. Heparin is an acid mucopolysaccharide. It acts by complexing with antithrombin. It acts by complexing with antithrombin to prevent blood clotting. Antithrombin is one of the natural physiological inhibitor of blood coagulation. Lithium heparin is the CLSI recommended salt of heparin to be used for laboratory testing. The sample collection tube stopper is color coded in green to indicate the presence of heparin. Heparin's interaction with antithrombin prevents coagulation. The interaction leads to the inhibition of thrombin. So we are now discussing about another type of anticoagulant, sodium fluoride. This anticoagulant is used for preparing blood specimen for the determination of glucose and urea in plasma by non-enzymatic methods. Fluoride inhibits glycolic enzyme and thereby prevent loss of glucose during transportation or delay in specimen handling. As fluoride is not a strong anticoagulant, it is mixed with Exolate. And this is a chart. You can see and read it. Thank you so much. Khuda Hafiz.